I'll give you 10 minutes. Essay on the board, right? Essay writing books, new people, right? Have a separate book for writing essays. Okay. Tunes. You have the tunes. Online people, anyone who didn't get the tunes yet? Anyone who didn't get the tunes? So don't keep this a habit, homework now homework. Okay. Class time again. Time then usually, okay? Because why? That sort of defeats the purpose, ne? Uh, I give you essays to write at home, not to, I mean, practicing essays is one purpose. The other thing is you have to sort of do a bit of repetition during the week, ne? නැත්තම් ආයි දැන් home. So today when you go home, so the fresh memory is very fresh. So it's going to gradually go down. You are going to forget things. But in the middle of the week, you are going to study a bit. So that decrease to zero will be stopped. Then you are going to fill it back up. Then it's going to come down. But because you have reinforced it one at least once, it's not going to go to ground level. Zero level එකට එන්නේ. පොඩි remnant එකක් it will remain, right? So when I think what I recommend is you write these things sort of Saturday night, no? Right? Saturday night the ආපහු it will come back bit up. Then it will never go down, hopefully, right? Because you have at least 3 times learned it, right? Then you're going to study it for the term test and some exam that we'll have in the class. So it will never come to zero level. That's the plan. Zero, you can't study the entire syllabus from a zero level in the last 2-3 months. At least, podi අර vague memory එකක්වත් තිබ්බේ නැත්නම් you can't do it. Okay? Only you're going to get stressed out. Okay? Right. Uh, so your marks are given like this, huh? 6 marks for the diagram of parenchyma, 6 for the diagram of collenchyma, 2 for sclerenchyma. That's how they give marks. That is, so you get a total of 14 out of 150, yeah? Right? Total out of 150. Uh, when you sort of narrow it down to 7.5, how much is that? Gana Dalaba? 150 becomes what? 150 marks becomes 7.5 out of what? 100. Paper again 100 dwelling 7.5 comes from one essay question, right? So this is like the average sort of amount of marks you get for a diagram, maximum 15 or maximum 20 out of 150, right? So narrow it down to 7.5. Someone like 14 out of 150 is how much out of 7.5? Calculate and see. Sometimes what I'm saying is always try to draw the diagrams, but sometimes if you are running out of time, it might not be valuable, worth wasting the time, right? At that time, you can go back and maybe some fill in the blanks in the structured essay paper. Write some more things in the essay paper, right? Depends, right? But you cannot, I mean, sometimes you are going to need these 14 marks as well. You can't, I mean, so that's the proper way or the complete way to write a essay question, but you have to be strategic about it, right? Time waste and other. If you are going to spend a lot of time, if you haven't practiced the diagrams, definitely you are going to need a lot of time to draw the diagram. So the best thing is to practice the diagram. Got it? Right? And be familiar with the labeling again and again. Right. So before in the revision class, I give you sort of like a complete PDF with all the like drawable diagrams. So there are Okuma diagrams in the resource. You don't need to draw. You don't need to be able to draw. Right? There are some diagrams you should be able to draw. Right. So I'll give you a compilation of such diagrams in your sort of when you're doing the paper classes and stuff. Right. So these three are such you should be able to draw diagrams. Right? Do I need to draw them in the board? We drew them last week, right? It's the exact same diagrams that you have in the resource, right? So let's start with the diagrams. What do you need to be able to draw in your parenchyma diagram? What do you need to be able to draw in your parenchyma diagram? Yes, parenchyma, what do you need? You should draw some isodiametric sort of spherical, right? Spherical cells with some good intercellular spaces, okay? With some good intercellular spaces and thin cell wall, thin cell wall. It has to be a thin primary cell wall, right? Thin primary cell wall. What else? What else? You have to have a big vacuole, right? Almost the entire plant cell is dominated by the big vacuole. and sort of nucleus is found sort of to the side, ne? Right? Nucleus is sort of found to the side. So what's the labeling? What are the things you need to label? You have to say thin, flexible. Yes, if you can say primary, come up, right? Thin, flexible cell wall. What else? Large central vacuole. Large central vacuole. What else? Okay. Intercellular space. Well, what is the mind? If you take a parenchyma, that's all, right? Yeah. Anything additional? No, right? Oh, nucleus. Yes, nucleus. Right, nucleus. So this is the parenchyma diagram. Collenchyma. Collenchyma, what do you need to do? Yes. You need to sort of thicken the cell walls a bit, ne? You need to thicken the cell walls a bit, right? Sort of polygonal with the candela, right? Sort of draw them in a sort of polygonal way and sort of draw the inner border of the cell wall like this. Okay. Then inside you have to definitely again draw a big vacuole, right? A big vacuole. And the thing is, sort of the cells are sort of closely packed together, right? So there is not many intercellular spaces, got it? So අපි සාමාන්‍යයෙන් when we are drawing plant cells, this is the typical sort of plant cell that we draw, right? සාමාන්‍යයෙන් with the boxy cell wall that we draw, right? So collenchyma sort of look like a typical plant cell with the sort of boxy polygonal cell wall. Okay, because why it is unevenly thickened in these sort of corners, right? What is deposits? What are the what I mean? What is deposited in these corners? It's still cellulose, right? Nothing else, but it's there's more cellulose. Why do you have more cellulose? Yes, because collenchyma are mainly involved in providing support. So what do you need to label? Something additionally, cytoplasm කියලා label එකක් දාන්න, right? Vacuole. So vacuole is there. What do you say? Thickened corners of cell walls. Thickened corners of cell wall. That's very important. Thickened corners of cell walls, vacuole. What else? Cell wall label එකක් දාන්න, right? This is the cell wall. Nucleus ඕනේද? Yes, nucleus and cytoplasm. Right? Cytoplasm. හරිද? Collenchyma, there's a bit more labeling, right? So coming to sclerenchyma, it's interesting because you don't need to label it, right? Label can there's no need, right? And also the only diagram that you have in your resource is the diagram of a scleroid, ne? There's only a scleroid diagram, so no need to draw sclerenchyma fibers. Also draw some these sort of branching pits. Okay, the pits are sort of branching. No need to label, right? No need to label those. Okay, for this unlabeled diagram you are going to get 2 marks. හරියට ඇඳලා තියෙනවා. What are the things you need to draw? Thick cell wall, mother lumen and branching sort of pits. හරි. So these are the 3 diagrams. So I'll read out the points that you need to have written. Online people, you wrote the essay, right? Okay, so ground tissue consists of three main types of cells. මොනවාද? parenchyma, collenchyma, sclerenchyma, three marks. 1 mark each. එක දෙක තුන. ඔය තුන ලිව්වා 3 marks you are going to get. Okay. Okay, mark your essays. Mark your essays. Parenchyma has primary cell walls. 1 mark. For saying parenchyma has primary cell walls, 1 mark. Which are thin. Thin. Thin කියන එකට 1 mark. Thin. Okay. If you have written thin, you are going to get 1 mark. Large central vacuole, 1 mark. All together, දෙකම එකට ලියලා තියෙනවා නම් all together you have to write large central vacuole, 1 mark. Then some contain plastids such as leucoplasts and chloroplasts. 1 mark. Anything like plastids are contained, or if you have written leucoplasts or chloroplast. Any of those three, you are going to get a mark. Okay. So that is actually it. I mean, if I wrote that essay, there are so many other things I need to write. I mean, I would have written, right? They retain meristematic ability. What else? They are living at functional maturity, but they have not been given marks. Right? Doesn't matter. A logical person should always write those, right? It doesn't matter, you're right? So they have given marks filtered out, right? Because they are going to arrive at 34 points only. Okay, that's the trend. They are, so the earlier the pattern was what? You are going to have marks out of close to 50. 50 you are going to multiply by 3 and give out of 150. But now the trend is they are reducing the number of points. So the amount you have to write is actually low. But the amount you have to write is actually the same, because if you want to own the full marks, you have to write everything. Got it? Write everything about parenchyma, you know. Then by chance you are going to have everything in the marking scheme within that, what you wrote. That's the only way to score. Got it? Right. Then come to collenchyma. Collenchyma are elongated, 1 mark. If you have written elongated, 1 mark. Have only primary cell walls, 1 mark. Collenchyma also have only primary cell, which are thicker than parenchyma cell walls, 1 mark. Thicker than parenchyma cell walls, and next point, unevenly thicker. Unevenly thicker. That's all about collenchyma. ඔච්චරයි marks දීලා තියෙන්නේ. Okay. Then sclerenchyma, right? Sclerenchyma have secondary cell walls which are thickened by large amount of lignin. On mark. Sorry, 2 points. Thickened, then large amount of lignin 2. Okay. Thickened කියන එකට 1 mark, large amount of lignin 1 mark. Then two types of sclerenchyma. Okay, as sclerides 1 mark, fibers 1 mark. Then sclerides are irregular in shape, 1 mark. Sclerides are irregular in shape, 1 mark. Then sclerides are shorter and wider. කියන එකට 2 marks. Shorter 1 mark, wider 1 mark. Okay. Then Fibers are long, 1 mark. Fibers are long, 1 mark. Next, slender, 1 mark. Next, tapered. Are we still talking about fibers? Fibers are slender, 1 mark. Tapered, 1 mark. Okay. Then you come to the overall functions, right? So this is the structure that we described. Then you come to the functions, right? So you can go in those two ways. You can write parenchyma, then give some functions. Write collenchyma, give some functions. Write sclerenchyma, give some functions, right? Or you can do it. I mean, the way I'm reading out, you can describe the structure first, then go to the functions. එහෙම එන්නත් පුළුවන්, right? Doesn't matter. Okay, which way you are going to write. So the next point, they are talking about again ground tissue fills the gap between dermal and vascular tissue. Ground tissue fills the gap between dermal and vascular tissue, 1 mark. Forms cortex and pith, 2 marks. Forms cortex, 1 mark, pith, 1 mark. Mataka idea what is cortex and pith? If you don't, it's fine. Today we are going to learn. Na? Then come to collenchyma, sorry, parenchyma functions. Okay, come to parenchyma functions. Photosynthesis, 1 mark. Parenchyma does photosynthesis skill if you have written 1 mark. Then short distance transport, parenchyma functions, okay, 1 mark. Short distance transport. Then metabolic functions, 1 mark. For the word metabolic functions, 1 mark. Such as synthesis of organic substances. Synthesis word you are not going to get marks, okay. Without the word synthesis, you are not going to get marks, okay. Then storage of substances, next point. Still talking about parenchyma functions, storage. In next point, wound repair, right? Wound repair. I would give marks if you have written they retain the ability to divide. ඒක ලියලා තිබ්බත් instead of wound repair. හරිද? දෙකටම ගන්න එපා. Right? Instead of wound repair you have written they retain the ability to divide. Then take a mark. Okay? So they haven't given marks to tissue culture, that sort of thing. Okay? Then collenchyma, right? Collenchyma, they say provide mechanical support. Right? Provide mechanical support. But I would, yes. So yeah, no. In the resource you don't have, right, photosynthesis, but in the marking scheme there is, right. I think I told you to write down, right, photosynthesis ke liya karne in the resource. Yes, yeah. Synthesis of various organic products. So what is the main synthesis that you can see in a plant? In the Calvin cycle, you are making food, right? So by synthesis, that's what they are implying. Wait. Don't you have photosynthesis? Yeah, no. There's yeah. You don't have under the functions of parenchyma. You don't have. Okay. So write down in the resource. Okay. Otherwise, you are going to miss again. Then collenchyma provide mechanical support. That's it. But I mean, I would recommend that you write the full answer. Right. They they are flexible and also elongate while providing support. That's why you have collenchyma. Natta you can doggala, you can put some serenchyma. Why are you using Collenchyma? Wherever you need a living support, right? You need support, but you need living support because it should not restrict the growth, right? Okay. Then sclerenchyma functions provide support and strength. That's it. Okay. Provide support and strength. Photosynthesis is given in the introduction. So you can, yeah, you can say there, that is there, right? In the introduction of ground tissue, right? Storage, photosynthesis, support, and short distance transfer, right? So who is doing all those mainly? Except the function support, it's parenchyma doing those. Photosynthesis, short distance transport, and storage. All those are done by parenchyma. Yes. Right. Okay. Clear, right? All good? Yes. Okay, let's come to today's lesson now, right? Let me know your marks out of 150. Online people, send me your marks. Okay. Okay, 150. How much? Everyone drew the diagrams, right? Okay. Start there. Okay. Practice the diagrams at this point. Okay. Online people, send me the marks. Okay. Right. So today we have to move on to talking about the primary structure, right? We are going to mainly talk about vascular plants, right? This lesson is mainly focused on vascular plants. You know this, right? Proper tissue organization is found in vascular plants. So that's what we are going to talk about mainly in this lesson. So come to page number 116, right? Taggalender, page number 116 in the resource. Okay. So plant growth highlight involves irreversible increase in dry mass associated with the development of an organism. Irreversible increase in dry mass associated with development, right? Found the page, right? 116, na? Okay. Associated with development. Otana, the word, highlight the word, highlight the whole thing and put a box or do whatever around associated with development. Very, very important. No? When you're writing what growth, if you haven't written, growth is the increase in mass that is associated with development. There are increases in mass that are not associated with development, such as what? Okay, that's serious topic, right? Cancer, increase in dry mass. What else? Just eating food, right? Kilo ek vitara kama gawa, that's an increase in mass. But is it associated with development? No, it's a temporary thing. If you haven't go to the toilet in like a week, that's an increase in mass, right? That's is that development? No. Okay. Growth is always associated, should be associated with development. Plants also get this massive goals, right? Tumorous growth. So those are not developments, sorry, growths, right? They are sort of pathogenic, right? Okay, highlight that. Often it is associated with the increase in cell number as a result of producing more cells from the meristem, accompanied by elongation. Plants continue growth throughout the life, known as indeterminate growth, right? Unlike us, plants usually have their growth process continuing throughout their life, right? Usually plant form is gets limited by sort of physics, right? How tall they can grow start will start to be dependent on how strong their stem is, right? How how I mean what are the nutrients available in the soil, rainfall. So usually their growth will be limited by those things. Got it? A tall tree would stop growing taller because its stem is not what strong enough. In Nattang, you can't see that many tall trees because why? All the tall trees have fallen down, right? So the environmental factors start to limit their growth potential. Got it? But as humans, we have a limited growth that is mainly determined by our genetics, right? Usually big trees, woody trees, their sort of growth is limited by environmental factors, lack of nutrients and other sort of, got it? Right? Right. So we'll talk about the primary structure in two aspects, that is the root and the stem. Root and stem. In what? You have two categories under both, monocot and dicot. Okay. That's what we are going to talk about. Primary, under the primary structure. Okay. So I have given you some diagrams. Hands up who watched the short clip I sent to the group. Right. So that was just the root, right? I said. Root is the idea. Ballare. Right. Root එක කියලා දන්නේ right? Online people must have seen. Right? Did you see the YouTube short I sent you? Did you think it was from another channel? Never. No, right? I always forget about this blue color, right? So you can see the four diagrams in the your tooths, right? Take out your tooths. So they are the same diagrams that I used in the software. So you have a nice 3D idea. But ඔය ටික label කරලා විතරක් not enough, right? I'm going to I mean you're going to draw the diagrams by hand also, okay? Right? So you're going to draw the diagrams by hand for, I mean, all four diagrams, okay? Because the next lesson is going to be how those primary structures change into the what? The secondary form when the plant is going to be what we call as its mature form, right? When it starts to get wider. So that's the next step of this plant structure lesson, right? So let's read the resource book first, right? Primary structure of the root. Highlight, okay? Apart from the distribution pattern of xylem and phloem tissue structures, the tissue structures of both monocots and dicot roots are more or less similar. Okay, right? Okay, let's think about the diagrams later. Outermost cell layer is epidermis. Highlight. Outermost cell layer is epidermis. Some epidermal cells produce finger-like extensions called root hairs. Highlight root hairs. Epidermis protects the inner parts while the root hairs involved in absorption of water and minerals. Right? So two separate functions. What's a root hair? It's a again, it's a specialized epidermal cell. How big do you think is a root hair? How many cells? How many cells do you have in a root hair? Any guesses? Only one. It's a unicellular structure. It's a single cell extension that is there to increase the surface area, right? So even though we have drawn them like big like human hair එකක් වගේ long, right? They are like very small, like a fine bristle, right? නිකන් අර what we call as in singular like a බුවක් වගේ, right? It's just they are like it's like a fur over the surface of the root, right? Very microscopic, small, right? I think you have a diagram of a root hair in this in the diagram in the previous page. Here, can you see in this diagram? The root hair is coming from a single cell, right? The root hair is coming from a single cell. Yes. The previous page, this page number 3. Page number 3, yeah. Can you see in the bottom diagram? A root hair is composed of a single cell. Okay. So a root hair is a modified epidermal cell. Ari, what are the other modified epidermal cells that you have in the epidermis? Specialized, sorry, specialized. Okay, trichomes. What else? Last week, right? So not in the root, but in the stem, what do you have? Stomata, openings. Who is there to control the opening and closing of stomata? Guard cells, specialized epidermal cells, right? Those kind of things, right? Root hairs, guard cells, those are the specialized epidermal cells, right? Okay, keep reading. Epidermis protects the inner parts while root hairs involved in absorption of water and minerals. What is the strategy? What is it using? By increasing the surface area. If you like, you can write with a pencil. Root hairs increase the surface area. Because later we'll sort of study exactly how root hairs absorb water. For that, the greater the surface area, the more water you can absorb. Okay. Right. Between epidermis and vascular cylinder, there is a ground tissue known as cortex, which is made up of mostly parenchyma cells with intercellular spaces. Highlight cortex and made up of mostly parenchyma, right? Functions of cortex and ඊළඟට, right? Mainly stores carbohydrates and also transports water and minerals towards the endodermis, right? So that's what we are going to talk about next. Endodermis. After reading, I'll explain. Na? Innermost single cell layer of the cortex is endodermis. Innermost single cell, highlight single cell. Endodermis is single cell, whatever endodermis is. Okay, I'll explain. Right? Endodermis consists of a suberin belt, highlight, called Casparian strip, highlight, and no intercellular spaces. Therefore, it blocks cortical apoplast from vascular apoplast. Okay. Highlight no intercellular spaces also. No intercellular spaces. Okay. Inner to endodermis, there's a pericycle. Highlight the word pericycle. Pericycle. Two containing two or three parenchyma cell layers. Highlight two or three. Yeah. They can ask questions like this. How many cell layers thick is the pericycle? Two or three. What is the composition of the pericycle? Parenchyma. Right. Then these cells in the dicot roots have a meristematic function and involved in the formation of lateral roots and the secondary growth of the root. Highlight in dicot roots have a meristematic function. What has a meristematic function? Pericycle. Pericycle. Okay. So if you take like the single resource book, those have very fun names. What the peri? I heard in a recent single media. Okay. Inner to pericycle, okay, there's vascular tissue as a solid core. Xylem can be found in the middle, highlight. Xylem in the middle, and it is star shaped, highlight. Star shaped xylem. In what? In the dicot root, in the cross section of a dicot root. Phloem is located in the grooves between arms of xylem. In the grooves, when you are writing an essay, when you groove in a word, you have to write. Okay, you have to write the word groove. Okay, in the monocot roots, vascular tissue consists of a central core of parenchyma surrounded by a ring of xylem and phloem. Alternating xylem and phloem. Highlight the word alternating, ring of alternating xylem and phloem. That's something you have to write when you're writing an essay. Pericycle in monocot roots is not meristematic. Why is that? Do monocots undergo secondary growth? No. Monocot plants do not undergo secondary growth. You know this, right? They only have primary growth. That's very important. Very commonly questioned MCQ. Pericycle is not meristematic in what? Monocots. Right. So before we do anything, you need to know how things are transported in a root from the soil to what? What you have in the middle, right? So come to page number 3, that previous diagram I showed you. This diagram, the one on the bottom. So that shows how things are transported from the, let's say, the edge of the root. What is the edge of the root? That is the epidermis, to the inside. That is the what? What is the innermost layer? Vascular cylinder, right? So you can say, so there's an epidermis like epidermis has specialized cells. So epidermis is a single cell layer thick, no? Right? And epidermis has this sort of root hairs. Are there? Okay. So this is a root hair cell. Okay. Then there are other epidermal cells like this. Okay. Then in the middle, what do you have? In the middle, you are supposed to have your vascular cylinder that has xylem and phloem. Right? So I'll just draw a rough one. This is a star shaped xylem. This is X xylem. Right? Where is the phloem? We'll draw a proper diagram. Okay. I'm just introducing you this. These are phloem, phloem, phloem. What did we say? In the arms of xylem, right? So then around this you have our bicycle වගේ pericycle. Okay, 2-3 cell layers thick. හරිද? Pericycle. Okay, then we come to the most important person. Okay, that is your what we call as the, don't have enough colors, right? That is called the endodermis. Okay, endodermis. Okay, actually endodermis is how thick? Only one cell layer, but I'll draw it bigger so you can see it. Right, it's only one cell layer thick. හරිද? Just to understand its structure, I'll drawing it bigger. Okay, හරිද? Right. So endodermal cells are like this. There are endodermal cells surrounding what? Root vascular cylinder. So it has cell walls like this. It has cell walls, ne? It's a plant cell. Cell walls. These are unnecessary, right? Just focus on these. So in between the epidermis and your vascular cylinder, right? What do you have? Okay, there's a ground tissue that is called the cortex. Cortex is composed of what? Mainly parenchyma cells, right? So those parenchyma cells have also their sort of their cell walls. They have their cell walls. They are sort of like arranged like this, right? Okay. So they have also have over there cell walls, right? So how is water transported from this sort of a root here up to this vascular cylinder? I'll draw the path there. So this is a water molecule that is in the root. It is going to be absorbed, right? Most of that water is going to be absorbed into the cell wall. It's not going to go inside the cell, right? Why? Going inside the cell means what? You have to come to the cytoplasm. To come to the cytoplasm, what do you have to cross? Plasma membrane you have to cross. Plasma membrane is a selectively permeable membrane. So there's some resistance. No? Imagine it's like a road without any gate, and there is the highway with the toll gate. You are not allowed, or everything is not allowed. But you have to slow down there, right? It's not like entering a road that is not having any gate, no? Right? So if you want to enter the highway, you have to sort of slow down. No? So the going through the plasma membrane is bit more resistant, slow. So water is not going to go through all these plasma membranes. It's only going to go through all the cell walls. It's going to go through the cell walls because why? Cell wall is like a matrix composed of cellulose. It's like a clear path for the water to go, right? So water is going to follow this pathway. These pathways we are going to learn in detail. This pathway of water going through cell walls is called here. Have you heard? Did you have this in the O levels? No, right? It's called apoplast. It's called the apoplastic pathway. So these are two adjacent cells, right? How's the what is the pathway of the apoplast? It's not going to cross into the cell. It's only going to go through what? Cell walls. What do you have inside this cell? You have this plasma membrane, red color plasma membrane. Na? This is just an introduction. We are going to talk about this. So this pathway is called apoplast, right? If the water is going to cross this plasma membrane once, I mean, if it's going to cross it, and then what structure did I draw here? What is this channel? Canva between two plant cells? Plasmodesmata. Plasmodesmata. So that is also a pathway they can take, right? Because among these cells you can have plasmodesmata, right? That is called symplast. Okay. Then there's another pathway they are not going to go through plasmodesmata. They are just going to go through the cell walls, crossing plasmamembranes, crossing plasmamembranes, crossing plasmamembranes. Like they are not going through plasmodesmata. Three pathways are there, right? Apoplast, symplast, transmembrane. කියලා තුනක් තියෙනවා. Why am I teaching you this right now? Because it was just mentioned in the resource. So we'll learn about in detail later, right? So tell me, using general knowledge, what is the least resistant path? යන්න ලේසිම පාර මොකද්ද for water? This stop path through the cell walls, because cell walls are made of cellulose, which is sort of it can cellulose sort of adheres water molecules, so water can nicely go through the cell walls. Okay, what is the most difficult path there? Most resistant? Repeated crossings, transmembrane crossing plasma membranes again and again and again. That's the most resistant path. So that's not good, right? So what is logical thing? The water is going to do the apoplastic path, right? So that's the advantage of the apoplastic path. Why? It's the least resistant. Tell me a disadvantage. It's like what? Have you gone right the Marine Drive like at around 12 p.m.? It's like the highway, right? You can go at 150, 200 විතර වුණත් යන්න පුළුවන්, right? But what's the problem? There are three wheels, motorcycles, pedestrians crossing, right? Everyone is there. You have a normal straight road, very good. There are no gates, nothing, right? But there are problems in the road. There can be obstacles, not, I mean, bad things, right? So apoplastic pathway has no filter. Anything can go through it. What are the bad things for the plant? Toxins. Other sort of mainly toxins, right? Heavy metals. So all those are toxins. So those things can go through apoplast nicely, right? So it's fine. They come to here. They come until this orange color. What is the orange color layer? Yes, endodermis. It's fine until they come there. But this endodermis, it's like uh, it's like a checkpoint. It's like a checkpoint before you enter into the proper highway. They are going to filter out, right? That endodermis, it's going to put a stop to this apoplast. Then ඇති apoplast, right? Free flow is enough. It's going to stop the apoplastic pathway by how? What is the best way to stop the apoplastic pathway? You cross the, you block the cell walls, right? You block the pathway by blocking the cell walls. That's what you have to do, right? So how are they going to block the cell walls? They have these suberin sort of strips of suberin in there, what we call as what? Lateral walls, side walls, right? So I'm not going to draw them, but it's sort of is around the cell wall, right? If you take a 3D view, right, you can see in the tube diagram, it's sort of like a wrapping layer around the endodermal cells, right? So I'm only going to draw it like this. Otherwise, you're going to get sort of the diagram is weird, right? Apoplast will be stopped by what? A layer of suberin. What do you call that? Casparian strips. What do you know about suberin? It's a lipid. Huh? What is the feature of lipids? They are hydrophobic. Water can't go through lipids. So, what have you done? You have stopped the apoplastic pathway. So, what is the option for now? For the water now? Apoplastic pathway blocked. They have no option but to what? Cross into these endodermal cells, then go into the vascular cylinder. When they are crossing into these endodermal cells, what are they going through? Plasma membrane. Plasma membrane, they have to cross. So when you're crossing the plasma membrane means what? They are going to get filtered. The unwanted things can be filtered by the plasma membrane of endodermal cells. This is the story. So, what is it going to do? You understand what the resource book now said, right? There's a suberin strip called the Casparian strip that is blocking the cortical apoplast. So, this is the cortex, right? So, this is the cortex. So, in the cortex, right, no issue. The water and minerals came very freely. The Casparian strip is going to put an end to this free flow, okay, preventing cortical apoplast from entering the vascular apoplast. Okay, what is vascular apoplast? The xylem is part of the apoplast. We'll talk about that later. Okay, so that prevents this water from entering the freely entering the xylem, or you can say vascular cylinder. That is the Casparian strip. Okay, so this diagram has everything everything I drew. Okay, page number 3, bottom diagram. Highlight the 3 routes. 1, 2, 3 online people, right? Page number 3. Highlight chuti edutiyenne, right? Because I have included uh, yes, bit small, no? Yeah. For 2028, I'll make it bigger. Right. Highlight the 3 routes: apoplast, symplast, transmembrane. Okay. Online people, you understood, right? You can clearly see everything. Right. Apoplast, symplast, transmembrane. In front of this apoplast, right, draw an arrow to the side. Yes, least resistant. It's the least resistant pathway. So this is apoplast. Apoplast, symplast. In front of apoplast, right? Least resistant. Least resistant. Okay. So that's why water travels in it. No? So you can see in that diagram, apoplastic route is by this sort of pink line. The pink line, number 1 kilati. You can see it traveling through the cell walls. Then symplastic route is by a blue line. Right? Can you see? So it is going to travel through what? Plasmodesmata, plasmodesmata. Going through one cell to the other through plasmodesmata. Right? Now can you see the apoplastic route coming to an end when it comes to endodermis, the red color endodermal cells? What's going to happen? They cannot cross the cell walls because of the Casparian strip. They have to cross the plasma membrane and enter into the endodermal cells. Harid? Then highlight endodermis controlled entry to the vascular cylinder. Controlled entry. Nice filtration. Okay. Let's draw our diagrams. Bigger, no? Right. So what are we drawing here? The roots. The roots of what? monocot and dicot. What? Primary structure. Right? Always remember what you are drawing. So outermost layer. Can. Outermost layer is what? Remember, this is a primary structure. So what do you have as the outermost layer? Epidermis. Single cell layer thick. What do you have? You have specialized root hairs. Root hairs are there for what? To increase the surface area. Okay, root hairs. Then what's the next layer? You have the cortex, right? You have the cortex. Inner to that, what do you have? Yes. What is the innermost inner layer inner to the cortex? Endodermis. It's like a checkpoint, right? How many cell layers thick? Only one. Only one cell layer thick. Right now, can you see it's up to here? The Ekama diagram I'm drawing, right? I'm drawing the same diagram, right? Don't mind if it's not properly circled because roots are not proper circles, okay? Right. Okay, right? Then what is inner to that? Yes, you have a pericycle. You have a pericycle that is 2 to 3 cell layers thick. You have a pericycle that is through 2 to 3 cell layers thick. Right. So up to here, everything common. So now I'll do the labeling. On this side, I'm drawing dicot root. On this side, I'm drawing monocot root. Okay. So what are the differences between monocot and dicot? Draw the dicot first. You have a star-shaped xylem. You have a star-shaped xylem like this in the center. Xylem. Okay. Star-shaped xylem in the center. Your phloem is on the sides, or you can say in the groove between arms of xylem. In the groove between arms of xylem. Okay. When you come to monocot, you have alternating rings of xylem and phloem. Alternating rings of xylem and phloem. So your xylem is sort of like this, sort of one column of xylem, whatever, right? Columns of xylem. Can you see in your resource, right? So these are xylem. Then you have your sort of phloems in between xylem. And in the middle, you have a core of parenchyma. Okay. That is the monocot root. Simple, right? So let's do the labeling. Yes, root hairs are there. Root hairs. Then epidermis. Epidermis. Then එළින්දල ඇතුළට යන, right? Cortex. Then after cortex, yes, what do you have? Endodermis. Orange color endodermis. Then what do you have? Pericycle, black layer. Then what do you have? I don't need to, I mean, okay, xylem. These are xylems, okay, and this is xylem here. Then phloem. These are phloems. Then you can say there's a core of parenchyma in the middle of monocots. Core of parenchyma. Right. Quickly label top two diagrams. Tooths, huh? In the tooths, top two diagrams. Identify first. Identify. Which side is dicot? Which side is monocot? Which side? This side. This is what? Dicot. This side, monocot. Then do the labeling nicely. Online people do the labeling. Then after you have done the labeling, come to the last page, blank page, primary structure of root, draw the two diagrams. How do you manage your space? Chutier and Chutier means like sufficiently large, but also manage your space. Online people got it? Any questions you have? Any questions? No? Be careful when you are labeling, right? So label in the tute first and then draw this diagram. Now, Medana, don't you have a question? What is that core of parenchyma? It's the pith, but don't write pith because why? Resource book has given it as core of parenchyma, but it is a pith, because why? What is the definition of core and pith? How do you, sorry, cortex and pith? The ground tissue outer to the vascular bundle or vascular tissue is what? Cortex. Inner to the vascular tissue is what? It's a pith. So it's a pith, but only label it as core of parenchyma. Okay. Quickly draw it. Label first. Label the tooth diagrams first, and then draw. Okay. So this sort of practice to draw the diagrams is essential for the next step that we are going to come. For secondary growth, you need to sort of have the idea of how the structures are located in the primary structure. Otherwise, secondary growth would be confused, right? Secondary growth is going to look like a load of circles, round, 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 ගොඩක් තියෙන්නේ secondary growth. I remember when I was doing A levels, කතා කර කර හිටියා කියලා in the school, right? I was brought to the front of the class and Andindiko secondary growth. So while you are there, you are going to get confused with all the circles, right? Because you have only one color, no, it's a black color whiteboard marker you are going to get and you have to draw, right? Even you are going to get confused with, okay, what cell ray is this, right? So this understanding and practice is very important for your secondary growth. In your school, what are you doing these days? Which lesson? Always in bio, ටිකක් මැඩම්ට කරදර කරලා හැම වෙලාවෙම bio lab එක ring and try එකක් දෙන්න. There are some good sort of these laminated specimens of certain slides, right? Uh, that you should try to observe. Okay, certain, I mean, like preserved laminated slides. Okay, bio lab එක ring කරලා try to do some that sort of thing, right? Otherwise, I mean, වලිදාගන්න එපා bio madam එක specific, right? Because you are going to at least do the practicals with them. Okay, bio and physics shape එකේ තියාගන්න ටිකක් because you are going to need to able to do the practicals. Okay, particularly physics practicals ටික අනිවාර්යෙන්ම do hands on at the physics lab. Because physics practicals are fun, right? You have this sort of a angular momentum practical, right? Holding dumbbells onto a side. You have done angular momentum, right? You bring the dumbbells closer and you start to spin fast. ඒක කළද ඒවා? පන්තින්න හැම එකම අර කැරගෙන පුටුවක් උඩට වෙලා ඩම්බෙල්ස් නේ මොකක් හරි ගඩොල් දෙකක් ඔය අල්ලගෙන right? කරකොල බලන්න, right? When you get them close, they are going to spin fast. Then up or slow. What do you call that? Conservation of angular momentum. Yes. That you have a very good stool in the lab, very friction free stool එකක් තියෙනවා. Right? If you take like a normal rotating chair, it's very, there's friction, so you are going to lose momentum. No need to draw this one, no? Okay? No need to draw. Online people, no need to draw this one. If you want, I have sent a photo to the group, so if you want to keep it, you can. But no need to draw it. Who's still drawing? I'll give you 2 more minutes. Online people still drawing? While you're drawing, think about it, right? In this dicot, it's X-shaped xylem or star-shaped xylem located in the center. There is no pitta or there is no core of parenchyma. If you take the dicot, okay, dicot root, there is no core of parenchyma. That is important. Okay, let's talk about the stems, huh? Right? Come to the resource book. Yes. Okay. Come to the resource book. Primary structure of dicot plant stem. Okay. Page number 117. Okay. Let's read it first. Outermost epidermal cell layer protects the inner parts from desiccation and infections. Highlight. So there are some additional functions for the epidermis of the stem. Okay. Online people, highlight the resource. Okay. Page number 170. In your stem, the epidermis protects additionally from desiccation. What is desiccation? Water loss, dehydration. Because why? The stem is in the outside, no? Right? It is exposed to sun, wind, and other things. So it can get dried out. So it has to protect from desiccation and also infections. Most of the insects, fungal infections, bacterial infections, all those happen usually to the shoot, right? The stem. So epidermis has to provide some protection from that as well. Epidermis is interrupted by pores called stomata. Highlight. So primary structure of the stem has stomata. Very, very important. Because you are going to lose that stomata when you come to the secondary row. So primary structure of the stem has stomata. Inner to epidermis, there is a cortex, highlight, containing mostly parenchyma cells. Collenchyma cells may be present just beneath the epidermis to provide strength. Highlight, collenchyma may be present. Sclerenchyma such as fibers are also present in the cortex to provide additional support. there can be some sclerenchyma fibers as well. Because why? This is a stem, no? So the stem should be upright. It should be a nice, rigid, or let's say a firm structure. The root doesn't have to be that. The root should be always flexible. Why? It should be able to sort of go through the soil particles and reach the groundwater and minerals and everything. Right? The stem should be sturdy, should be above the ground nicely. Okay. Then vascular bundles are arranged in a ring. Highlight. What is this stem? Dicot stem. Vascular bundles are arranged in a ring. Vascular bundle contains primary phloem towards the cortex and primary xylem towards the pith, and in between a cambium tissue. Okay. What is a cambium? What is a cambium? It's a meristematic tissue. What type of meristematic tissue? A lateral meristem. Okay. There are two types as vascular and cork cambium, where here actually this cambium tissue is vascular cambium. Okay, you can write යරට මේකක් දාලා vascular cambium tissue. Okay. Outside the vascular bundle, there's a cluster of sclerenchyma. Okay. Outside the vascular bundle, there's a cluster of sclerenchyma that we call as a sclerenchyma cap. It's like they are to sort of provide protection to the vascular bundle. Okay. Inner to vascular bundles, a large pith, highlight, which is made up of parenchyma cells can be found. That is a large pith. Okay. Lateral shoots develop from axillary buds. It's not relevant there. Okay. Then come to monocot stem. Monocot stem. Right. We are only going to talk about the differences. It has a epidermis. Okay. Right. Epidermis is there. Then what else is there? There is stomata in the epidermis. So those are common things. We are not going to mention them again. Then ground tissue of monocot stem is not differentiated into cortex and pith. Very, very important. Highlight. It is then what do you call that? It is just ground tissue. It's just ground tissue without any differentiation into cortex or pith. Vascular bundles are scattered throughout the ground tissue in most monocot stems. Scattered throughout. There is so randomly sort of everywhere, not arranged in a ring. Each vascular bundle is surrounded by sclerenchyma. Highlight. It consists of a xylem and phloem, but no cambium. Highlight. There is no cambium. Because why? Monocots don't undergo secondary growth. Cambium is there to do what? Secondary growth. Highlight. No cambium in between. Okay. So the stem, in your syllabus, you have only the secondary growth of the stem, but you mention a bit of the root secondary growth as well. So pay attention to the primary structure of the stem. Right, there is not much. Did you have it in O levels? This vascular bundles arranged in a ring, that story you didn't have, right? Okay. Dicot stem and monocot stem. So as always, what do you have? Outermost layer is what? You have an epidermis. Epidermis. But does that epidermis have root hairs? No. Because why? This is the stem. Okay. So it is again usually a single cell layer thick. Providing protection from, you can say, pest attacks and in additionally desiccation. How is it mainly protecting from desiccation? Anyone? How is it mainly protecting from desiccation? Hmm? With the cuticle. There's a layer of wax called cuticle. What is it composed of? Cutine. It's shiny and waterproof, right? So it can reflect excess sunlight a bit, but it's also waterproof. The cuticle thickness can change. It can have, some plants can have a very thick cuticle depending on their sort of habitat. Okay, if they are exposed to a lot of sunlight. Epidermis. Then what do you have? Let's draw the dicot stem. In the dicot stem, what I say is sort of draw a small, this sort of a dotted line. That is the vascular cambium ring. Okay. Then what you do is I'll stick to what you have in the resource. You draw your vascular bundles like this. Draw at least 6 or something, right? 1, 2, 3, 4, 5, 6. Draw at least 6. Hatarak is not enough. Draw at least 6. Right? Vascular bundles are arranged in a ring. Okay. So now draw what do you have in the middle? Yes. You have vascular cambium. Right? Vascular cambium. So who is to the outside? What is this? Outer to the vascular bundle, what do you have? The cortex. What is towards the cortex? You have your phloem. Phloem is to the outside. Xylem is to the inside. Then you have a sort of a sclerenchyma cap. Everybody hat or a helmet. You have a sclerenchyma cap. It is to give some protection and some sort of support to the vascular bundle. This is the dicot stem. Very simple. Come to monocot stem. There is no such ring arrangement. They are randomly scattered. Right? Randomly scattered. Vascular bundles are randomly scattered, sort of like this. There is no differentiation as ground tissue. Sorry, as cortex and pith. Just the ground tissue is there, right? No need to even label the xylem, right? So usually your xylem can be like that. No need to differentiate the xylem, right? And around them, also no need to draw a xylem, sorry, a sclerenchyma layer, right? They usually have a sclerenchyma layer around each vascular bundle. Okay, I'm going to stick to the diagrams that you have in the resource, right? Simple diagrams. So do the labeling. What do you have? Yes, outermost layer. Do the labeling with me in your tube diagrams. Online people, page number 4, bottom diagram. Right? Okay. Epidermis. outermost layer. Okay. Then what do you have? Cortex. Cortex. Go from outside to inside. Then sclerenchyma fibers do a labeling. Sclerenchyma fibers මේ පැත්ත විතරයි. Sclerenchyma fibers. Only in your dicots you have to do that labeling. Cortex. Then you have phloem. Phloem. Then you have what? Cambium. Orange color cambium. Okay. Xylem. These are, to be more precise, these are what? These are primary xylem. This is primary phloem. Okay. හරිද? Then what is in the center? I'll draw right here. This is a pith. Right? And there's also, I didn't draw it. There are some things called vascular rays. There are vascular rays that are sort of connecting the cortex to the pith. Okay. So label them. Ground tissue, they are called vascular rays. Okay. Ground tissue connecting cortex to pith. Here in the monocot, it's very easy. These are all vascular bundles. Everywhere else you have ground tissue. Did I miss any labeling? No. If you can, මෙහෙම එකක් දාලා, this is what? This is a vascular bundle. Any issue with labeling? This one? No, right? What is this side? This is dicot. This is monocot. That is simple. Then you can see epidermis, you can see cortex, you can see pith in the center, you can see. You can see the sclerenchyma cap. In this diagram, it's sort of pink colored. No? Sclerenchyma fibers. Yes. Then blue color phloem layer is there. Then light blue color vascular cambium is there. Then inner yellow color xylem is there. On the opposite one, so actually in these vascular bundles, there are xylems and phloems. You don't need those, right? Just label it as vascular bundles and the ground tissue. That's all. Okay, draw this, huh? Nicely, dicot, draw it below, nicely below the dicot root, huh? Dicot stem, monocot stem, huh? Other people, any questions? If you can't see the board nicely, I sent some photos to the group, huh? Check it. Monocot diagram. Ah, yes, sorry. Yeah, good. Huh? I have labeled cortex here. ඔගොල්ලෝ දැක්කේ නෑ? Right. There is no cortex. Right. Exactly. Thangala blame එක දාන්නවා, right? You didn't tell me, right? Okay. There is no cortex in the monocot. So while you're drawing, listen to this. Huh? So we are going to start your paper class. Okay. You're starting your paper class. The date I decided is Wednesday. Any issue? Wednesday, 7 to 9. Hands up who can't. You have another class. When does it finish? 7 finished. 7:30? Online, no? It's happening online. Finishing at 8. 8. Isn't it too late? Issue there's no such issue. I'm fine because I'm going to be doing it from home, right? So, right, I didn't confirm. So, 8 to 10, fine then. Anyone? Too late? Online people? Wednesday, 8 to 10. The higher start. Oh, the higher නිදාගන්න. No issues. Okay. Okay, I'll put a poll, right? I'll put a poll in the group. We'll see. So it's going to be like this. For you physical people, I'm going to create a separate group for the paper class, right? People who are joining for the paper class, and I'll put the link there. You have to pay the fee to the office down. Okay. So the combined fee would be, if you're coming to both theory and the paper class, there would be a discount. That's the normal way. And we are going to mark your papers and give, right? So we are going to mark your papers. I plan to do 2 papers per month. MCQ 1 week discussion, then SCQ or the second part another week discussion. Right? We are going to do 2 papers per month and they will be marked and given. That's how the paper class is going to go. Online people, it's usually the same. In the website, you have a 2027 paper class and February month free. Yeah? We have 2 more days. No? February month free. You don't need to pay. You can join. Okay. March onwards, it's going to go as normal, right? What's happening? Message na hoga. What's the duration for the paper marking? What do you mean? What about Tuesday? No, I'm starting a class in Nigambo for the 2028. Until 9 p.m. you have a class. Class from 9 to 11. Physics from 8 to 11. Chemistry class. Wednesday is issue again. I'll put a poll with the dates I can and we'll decide impartially. Yeah. Okay. You can, I mean, if you have friends, I mean, they can just join just the paper class also, right? They don't have to come for theory. They can join the paper class only that as well, right? They can send the papers and we can mark and give you. Class till 7. Yeah, I'm going for online students. For everyone, I'm going to send the PDF and you have to send the PDF to me. Right. For physical students, I'll give you papers printed. Right. And I'll decide on a paper writing slot as well. Right. Then you can hand over the papers to the office and we'll mark. Okay. After you have drawn this, you can take a break. Huh? Be careful with the labeling. Okay. Don't forget to label ground tissue connecting cortex to pit because in secondary growth they are going to play a role. Huh? These green color lines that are connecting the cortex to the pit. Okay. Don't miss the labeling. What's going to happen if you miss the labeling? One labeling in a diagram, half marks. Okay, half marks. You miss at least even one labeling. Right. Okay. So we better do some questions, no? Right. Okay. So in your question, Tute, do we have anything that we can do? No, right? No MCQs, particularly from primary and secondary growth. Okay, right. Shall we move on? No questions from the primary stem and root, right? So come to your resource books. Online people, can you hear? Yes. So we can start secondary growth. Okay, we can start secondary growth. There's a question. If cambium involves in secondary growth, how can primary dicot stem have vascular cambium? That's because it's already there for the upcoming step, right? Vascular cambium is already in location or already in place to do the, I mean, its future function. What is its future function? To do secondary growth. We are going to do it, I mean, we are going to learn about it in a few minutes, right? It's already there because it has the potential to do the secondary growth. So it's just in place in the primary stem because primary stem is going to undergo secondary growth in the dicot, right? But in the monocot, you don't have such a cambium because why? They are not going to do any secondary growth, right? Okay. Okay, page number 118, secondary growth. Okay, very, very important. So pay close attention, right? Definition. Increase in diameter of stems and roots in plants due to the new cells produced by lateral meristems is called secondary growth. Lateral meristems. Highlight that one more. I mean, right? Lateral meristems. So who is causing the secondary growth? Your lateral meristems. This occurs in stems and roots of woody perennial plants, including all gymnosperms and many dicots. Highlight woody perennial plants, including all gymno highlight, very, very important MCQ points, and many dicot species. Not all dicots, but many dicots. Okay, not all dicots, but many dicots. Next bullet, right? Lateral meristems, namely vascular cambium and cork cambium, produce cells and tissues in secondary. What are the two cambiums? Lateral meristems, vascular cambium and cork cambium. Highlight. Then vascular cambium adds secondary xylem or wood. Highlight. Secondary xylem is known as wood towards primary xylem and secondary phloem towards primary phloem, increasing vascular flow and support to the shoot. So what are the tissues produced by vascular cambium? There is already primary xylem and primary phloem. Who is it going to produce? It's going to produce a secondary xylem and a secondary phloem. That's it. Okay, we'll see how all that tissue is originated, right? Then the cork cambium. What is it going to produce? It's going to produce a thick, tough covering consisting mainly of wax impregnated cells that protect the stem from water loss and from invasion of insects, bacteria, and fungi. Okay, so what is it called? You can write an arrow there. This thick, tough covering that is produced by the cork cambium that is replacing epidermis. So it's going to replace epidermis. It is called the periderm. Periderm. So in your secondary structure, you don't have a epidermis. What do you have? You have periderm. Okay, next point. In woody plants, primary growth and secondary growth occur simultaneously. As the primary growth adds new cells and lengthens the stems and roots in the younger regions of the plant. A secondary growth increases the diameter of stems in all the regions where primary growth has ceased. What did you understand from that? It's going to happen simultaneous, right? What do you mean by primary growth? Usually lengthening, elongation, growing taller. Okay, so the shoot is going to grow tall, root is going to elongate, ne? Right? So imagine there's like a there's a massive tree, right? Is it going to do only primary growth? Then it's going to become wider, no? Right? While it is growing taller, it's getting wider. While it is growing taller, wider. It's going to happen simultaneously in woody plants, right? That makes sense, right? You're going to build some more, then you're going to strengthen. Build a bit, strengthen it. Right? So first, what's going to happen? Primary growth, that is going to do the lengthening. Then you reinforce or give strength to that grown part by the secondary. Simple. Highlight, primary growth and secondary growth occur simultaneously in woody plants. Secondary, okay, next page, right? Let's read it and let's draw a diagram. In your tooths also, I have given you some clear diagrams. So have it on the side as well, right? Have your tooths on the side as well. Page number 5. Okay. All right, people. Let's read. Secondary vascular tissue is produced by the action of vascular cambium. What are secondary vascular tissues? Secondary xylem and secondary phloem. In a typical woody stem, the vascular cambium consists of a continuous cylinder of undifferentiated cells, often a single cell layer in thickness, located outside the pith and primary xylem and inside the cortex and primary phloem. Okay. So if they ask you to describe the vascular cambium, how you describe the orange layer? Okay. It is located, yes, only it's a single cell layer in thickness. It is located outside the pith and primary xylem and inside the cortex and primary. That's how you describe. It's undifferentiated loop. Why is it undifferentiated? Because it's meristematic tissue. Meristematic tissue is undifferentiated. Cambium is a meristem, right? Sorry. Then in a typical woody root, okay, we'll talk about the root secondary growth later. So ignore that, right? Skip that. Typical woody root, the vascular cambium forms laterally exterior. That we'll talk later. Mulima, let's focus on the stem. So forget that for a bit. As these meristematic cells divide, they increase the circumference of the vascular cambium and also add secondary xylem to the inside and secondary phloem to the outside. That makes sense. No? What do you think? Here is the cambium. It's going to add more cells of xylem towards the inner parts of the root, more phloem to the outside, because that's how it's orientated, right? Xylem to the inside, phloem to the outside. Okay. Viewed in cross section, the vascular cambium appears as a ring of initials. What do you mean by initials? It's like, I mean, so there are meristematic tissues, meristematic cells. Those meristematic cells are the first step of whatever they are going to become. They can become. So if they are xylem, what are they going to give rise to? Xylem, vessel elements, tracheids, parenchyma, and fibers. So all those, I mean, in this vascular cambium, you can see the sort of beginning of formation of those 4 types of cells, right? That's what you call as initials, right? A ring of initials. And also towards the phloem, you can see what? Tube elements, companion cells, parenchyma, and fibers of phloem, right? So those are what you call as initials, right? Some initials are elongated and are oriented with their long axis parallel to the axis of stem or root, right? So if I explain this, I'll erase this side. So depending on how of their orientation, we can have a idea of what they are going to produce, right? So in a stem, what is the given as longitudinal axis, right? Yes, axis. What is the axis of the stem? What do you mean by the axis of the stem? So if this is the stem, where is its axis? It's going through it. The straight line going through it, right? Through the center again. That is the axis. What do you mean by perpendicular? Like this, 90 degrees, right? So those two idea keep in mind, right? This is the axis and this is the perpendicular axis. Okay. So if you are going to produce xylem and phloem, what are xylem and phloem? They are transport tissues. So what is the direction of transport? How do you, I mean, you usually transport along the length, no? Right? Along the stem or the root. So if I complete this route, it's like this, no? This is the stem. So we are looking at a cross section of that stem. So if you draw an axis through this, it's sort of going upwards or downwards, right? Right? That is the axis of the stem or root. So if you take those initials, what are initials? Those are the are first undifferentiated. I mean, they are the sort of the origins of xylem and phloem. They are going to give rise to individual cells of the xylem and phloem. Okay. So you have two types of initials that you should. I'll explain right now. Okay. This side has more space. No? So I'll erase the secondary, the primary structure of the monocot root stem as well. You don't need that, right? Because it's not going to undergo secondary growth. So මෙයාව මම දක්වන්න පුළුවන් for now. We only need this one for now. Okay. We are only going to need the primary structure of the dicot stem only. Okay. So if you talk about those initials, originating from what? Vascular cambium. I told you there are two types of those. There are initials that are oriented with their long axis parallel to the axis of stem or root. So what is the axis of the stem or root? This one. So if there are initials in this vascular cambium that are in the that sort of long axis direction of the stem or root, those are called what? Those are going to give rise to what? They are going to give rise to the actual vascular tissues. So there are initials oriented parallel, no? Stem or it can be the root. They are going to give rise to individual cells of xylem and phloem. What are the individual cells? You have your vessel elements, tracheids, blah blah, right? And if you take sieve tube elements, companion of phloem, right? It's like this. So if this is your vascular bundle, okay? This is your vascular bundle. This is the vascular cambium, and in that vascular cambium, those initials are sort of oriented like this, parallel to the, what is the, so this is the axis. Now imagine the axis is going through the center, right? Got it? So that is the fate of initials oriented parallel to the axis. There are some initials that are short and oriented perpendicular. Okay, there are short and oriented perpendicular. Oriented perpendicular to the axis. So what do you mean by oriented perpendicular? So they look like this. So they are sort of 90 degrees to the axis. Okay. What are they going to give rise to? They are going to give rise to, they are sort of short connecting channels in between the cortex and the pith that are here, right? The ground tissue connecting cortex to and pith. It's going to sort of advance itself in the secondary growth, giving rise to what we call as vascular rays, right? The proper vascular rays you are going to get here. They're like a short, what do you say? Small communicating or like perpendicular connections between the cortex and pith. Okay. That's why we didn't call it vascular tissues here, because the proper vascular rays are going to come in the secondary structure. It's still here, it's just ground tissue. Okay, got it? That initials katha, I mean, you don't need to think about it that much. Or I need to give you the sort of the, let's say, organization of information, yeah, right? So read, other initials are short and oriented perpendicular to the axis of the stem or root. They produce vascular rays, which are parenchyma cells that connect the secondary xylem and phloem, store carbohydrates, and aid in wound repairing. We'll talk about these vascular rays when we are drawing the structure, okay? Right? Then keep reading. As secondary growth continues over many years, layers of secondary xylem accumulate. This secondary xylem is very, very important. Why is that? What is xylem composed of? Xylem, vessel elements, and tracheates. Those are dead cells, right? Very, I mean, they have thick lignified cell walls, which are very strong, right? What is the nature of wood? Wood is usually sort of dry and strong, right? It is resistant to digestion. We say like a good type of wood, it's resistant to even insects, right? Right? Because why? What do you mean by good type of wood? It has very thick lignified cells now. It's very resistant to degradation, which is a good wood, right? Or costly, right? They have a nice thick secondary xylem. That's what we are going to use as furniture. We are using secondary xylem as furniture. Okay, we'll talk about this wood in detail. Okay, so uh, the walls of secondary xylem are heavily lignified and account for the hardness and strength of wood. So secondary xylem is very durable. Okay. During early stages of secondary growth, the epidermis is pushed outwards, causing it to split, dry, and fall off. So, what did I tell you? There are cell layers being produced inside the stem. By who? Vascular cambia. Right? So, is this epidermis going to survive that? ඇතුළෙන් නිකන් හෝම් පිපිරෙන අර shirt එකේ buttons වලින් පිපිරෙන්න යනවා වගේ, right? When someone is getting very fat or something, right? And Harry Potter, that Aunt Marge in the third film, can you see her? Because he makes her expand. So can you imagine her button some right? It's like that. The epidermis cannot survive. It's not that stretchy. It's not very stretchy. So when new things are being filled inside, this epidermis is going to, what's going to happen to it? It's going to rupture at certain places. It's going to rupture. Split. Split. then epidermis will fall out. Helen. So what now? It's like a skinless stem, no? Hamak na, no protection. There's going to be a better skin, better upgrade that is more durable, more hard. That's going to replace it. What do you call that? Periderm, right? Periderm will be produced by who? Yes, cork cambium. Okay. Then next, read a cylinder of dividing cells arises in the outer layers of cortex in stem and pericycle in root, forming cork cambium. Matakada, when we are talking about the root, that pericycle had meristematic function. I told you, right? So that's how it's going to work, right? In the root, pericycle is going to produce the cork cambium, and in the stem, so this is the cortex, right? This is the cortex. In this sort of outer layers of the cortex, you are going to get a cork cambium. So cork cambium, what is it going to do? Produces cork cells to the exterior and to the interior. Okay, just read, then with the diagram I'll explain it. It's going to produce cork cells. Okay, cells added to the exterior become cork, right? In singular we call it the elate, right? Cork cambium and the tissues it produces are collectively known as periderm. Okay, if they ask you what is periderm, that is the definition. Okay, highlight cork cambium and the tissues it produces is called the periderm. As cork cells mature, they deposit a waxy hydrophobic material called suberin. We are facing suberin again. Cork cells have suberinized cell walls. Highlight suberin. The cork tissue function, hydrophobic material called suberin, and they become dead cells. So, cork cells, mature cork cells are dead cells. Okay, mature cork cells are dead. Very important detail. Cork tissue function as a barrier that helps to protect the stem or root from water loss, physical damage, and pathogens. Why? They are dead, right? So they are like a dead suberinized barrier that is going to protect the stem. Each cork cambium and the tissues it produces comprise a layer of periderm which is impermeable to water and grasses. Suberin is waxy, so definitely water can't go through. And these cork cells are tightly packed. So what is it going to do? It's going to make it airtight as well. Can the stem be survive like that? Watertight, airtight? No. So then what the stem is going to do is it's going to have sort of ventilation openings, right? For ventilation, it's going to have some openings, right? Those are called lenticels. Come to the next page, 120. For gaseous exchange, small pores are present in the periderm known as lenticels, which are formed by loosely arranged cork cells. Highlight the word lenticels formed by loosely arranged cork cells. I'll explain all this again, okay? Loosely arranged cork cells. They appear as horizontal slits. Remember, if you take a look at a go home, if you have a good tree at your home, horizontally on the tree, sort of the stem, you can see sort of slits again, right? Those are their ventilation channels of the stem, right? Lenticels. Okay. Next, further growth of the stem or root breaks the layer of cork cambium and it lacks its meristematic activity and they become cork cells. Okay, so it doesn't stop here. The plant or the tree is going to get bigger and bigger throughout its life, right? So this cork cambium is going to also feel the push now. So there was a cork cambium here. That cork cambium is going to feel the pressure now as well. It's being pushed from inside. What's it going to do? What's going to happen is the cork cambium also going to rupture, split. That causes it to die and become non-meristematic. So in order to replace the dead coke cambium, there's going to be a new coke cambium arising from the inside. Right? There's a new king coming from inside, like to replace the old one. Right? So a new coke cambium comes. What is the coke cambium going to do? It's going to produce coke cells. Those coke cells are going to produce a new layer of lenticels. Then what's going to happen to the old lenticel? It's going to fall out. From the tree trunks, you can remove these massive පොතු වගේ chunks, right? පොතු කියලා කියන්නේ ගහේ පොතුම අපි ගලවන්නේ ඇඟිලි දාලා, right? That's that is part of what? Those are part of the coke cells or the old lenticels. Sorry, old periderm. Old periderm you can remove as tadi potu, ගස්වල potu, right? So a new coke cambium is initiated inside, which produces a new layer of periderm. As new cells are added, outer regions of coke will crack and peel off in many tree trunks. Right? එළියම තියෙන coke පොත්ත, it's going to get removed. Because why? There's a new one coming from the inside. So finally, due to the tissue layers produced by vascular cambium and coke cambium, girth of the stem and root increases in secondary. Girth එක increase කරන්න තමයි, oh, we do all these things. Okay. Okay. Stop right there. Then we'll draw the diagram. Okay. While I'm drawing the diagram, you can observe this diagram, page number 5. You can see at the top, you only have what? The primary structure. Because why? Because at the tip of the what? Shoot apex, what is still happening? Primary growth, right? So there you only have the primary structure. Can you see the topmost diagram? That is still this, no? මේකනේ තාම එතන තියෙන්නේ. Right? Primary xylem, primary phloem, that's all you have. Right? Gradually, in the lower parts of the stem, you are going to start slowly secondary growth. Right? Who starts it? Can you see? Highlight in the second diagram, can you highlight vascular cambium? Then below that you can see first coke cambium. Very, very important. Highlight first coke cambium. What does that mean? There are many coke cambiums to come. පළවෙනි පරාක්‍රම බහුව. Right? That means what? There's another one to come. You don't give a name, right? First World War එකේ ඉන්නකොට you don't call it the first World War, but because another one came, you call it the first one, right? Got it? Why do you call it the first coke cambium? Because there's more to come. Got it? පළවෙනි මහින්ද රාජපක්ෂ කියලා කියන්නේ නැහැ, because there are no more මහින්ද රාජපක්ෂ to come. එහෙමනේ, right? So there is more to come in the, we call it first coke cambium. Because why? You are going to need more and more periderm layers as the tree grows. Got it? Then finally you have number 7 කියලා තියෙන්නේ. Can you see? Most recent coke cambium. In the below diagram, most recent coke cambium. That is the most recent one. Okay. Got it? Right. So now what we have to do is we have to draw some circles. Okay. So you have to practice. You have to practice how to draw this. What I recommend is you start with who? How do you start to draw the secondary structure of a dicot stem? You can start with vascular cambium. Start drawing with the vascular cambium. So what's going to happen is, when secondary growth is about to start, this vascular cambium is going to become a continuous ring, one continuous ring. The method, there are some gaps, no? Those gaps are going to be filled and you are going to have a continuous vascular cambium. That's the first step, right? Then what are you going to get? Vascular cambium, what does it do? Yeah. Vascular cambium, what is it going to do? To the inside, what's it going to produce? Yeah. Secondary xylem. And this is secondary xylem. To the outside, what is it going to produce? Always refer that one. Huh? What is the fate of the things that are there and here? What to the outside? What's it going to produce? Secondary phloem. Secondary phloem. Right? Then still you have the primary phloem and primary xylem. Huh? So here you have the primary xylems still. Right? The primary xylems are still here. They are going to get small and small. I mean, they are going to small and also very reduced with time because a lot of secondary xylem is being produced over the time, right? So you still have your secondary phloems. Huh? Sorry, I'm sorry, primary phloems. Huh? Maybe the primary phloems are still there. Okay. Pay close attention, right? Maybe if you sort of don't understand this, it's going to be difficult, right? Secondary growth can be a tricky topic. Huh? Harith, what are these? Primary phloem, right? So that is the job of vascular cambium. That's the cation, right? That is the job of vascular cambium. What is it going to do? It's going to produce secondary xylem and secondary phloem. Right? So who is the second cambium that we need to talk about? Yes, your coke cambium. Right? So let's draw the coke cambium. Coke cambium is not something that we have here. So I'll mm, use purple color, right? Where is the coke cambium arising in the stem? Read the resource book and tell me, right? Where is the coke cambium arising? Okay. From which part of the stem do you have the coke cambium arising? Outer regions of Ambunnadevi. We just read it. Where is the coke cambium arising? Outer regions of cortex. Outer regions of cortex, right? Am I correct? In the stem, outer regions of the cortex. So around in this part, no? Outer regions of cortex. So what is this? The coke cambium. Right. So coke cambium, what is it going to produce? What are the tissues it's going to produce? Yes. Okay. Coke cells, right? Coke cells are going to be produced to the inside and the outside. Ahmed? To be exactly true, right? The cells that is produced inside will produce something called secondary cortex, right? Secondary cortex කියලා එකක් තමයි ඇත්තටම හදන්නේ, right? But you don't need to know that. Just remember the vascular cambium produces coke cells, that's enough. Okay. So it's going to produce coke to the inside and the outside, right? Then let's draw the final layer. After the final layer of, so there's going to be a coke layer. Then you have the outermost layer. What is that? That is the, okay, the remainder of your epidermis. So I'll draw it in green color. the remainder of your epidermis that has been left behind. Right? It has been ruptured in several places. පැලිලා තියෙන්නේ. Right? This is what is remaining of your epidermis. What is remaining of your epidermis? It's dying, huh? Right? Yes. So those are the things you need to know. Okay. And I'll draw this outermost layer of the periderm. That is your what? The new skin, huh? The new dermal tissue, which is the periderm. So I told you it's going to be round tick, okay? Is the secondary structure. Right? So it's a matter of identifying the individual circles, and the labeling is the most important thing. Okay? Labeling is the most important thing. Right? So if I remind you again, what are the things you can see? Orange color, vascular cambium. What is it going to produce? It's going to produce, what is this? Secondary xylem. It is secondary xylem. Okay, I won't write. Okay, secondary xylem. Okay, what is this also? This is also xylem. What is it? Primary xylem. Are then what is the blue layer? It's phloem. Phloem, 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 phloem. What are these? This is primary phloem, right? This is still the remainder of primary phloem. Harid. Then these are the cork cells that are produced inside. So who is this? Who is this purple layer? Yeah. Cork cambium. Mhm. Actually, you call it, we are supposed to call it, there's a, so still there is a cortex, right? So you don't need that because it's not labeled in your resource book diagram. So I'm not going to label it. This is called actually the secondary cortex. Okay. No need to label that because it's produced by the cork cambium itself, right? Then you have finally the outermost epidermal layer. What is it? Periderm. Ne? Periderm. So periderm is this one which has lenticels. We'll draw the lenticels later. Okay. So you can, I mean, it's optional. You can draw the older epidermis as sort of the ruptured epidermis that is still falling in the process of falling off. Okay. Let's quickly label. Right. What is in the middle? Yes. What is in the middle? There is actually a pith, but because it's not labeled in your resource, I'm not going to label. Right? There is a pith. Okay. This is what? Primary xylem. What is this? Secondary xylem. What is this? Secondary phloem. What are these? Primary phloem. Okay. What is this orange color one? That is vascular cambium. I'm sorry. Yes, vascular cambium. All right. Orange color is the vascular cambium. Then coke cambium we already labeled. You can say this is what? The tissues produced to the outside, periderm. Yes. Periderm. And then those vascular rays. Vascular rays are there connecting the xylem to the phloem. I told you what is their orientation. What is their orientation? Perpendicular. So what is the axis that is parallel? That is going into the board. Ne? That is a parallel axis going into the board. What is the vascular rays? They are oriented perpendicular, like this. Okay, you can see them, right? So these are green color. Vascular rays that originate from initials oriented perpendicular to the axis. Right? Any questions? Better say this is coke. Huh? Better say this is coke. Better say it's the first. Yeah, it is the coke. Okay, the coke plus coke cambium. So I'll label the coke cambium below. The coke plus coke cambium is known as what? Periderm. The coke plus coke cambium all together is called periderm. So this is the coke cambium. All together, this is what? Periderm. I'll explain this again. Okay. Secondary growth is the rest is done by the action of lateral meristems. What are the two types of lateral meristems? Vascular cambium and coke cambium. Vascular cambium, what is it going to do? It's going to produce secondary xylem towards the inside. That's it. And sorry, the secondary phloem to the outside, phloem and xylem. That is going to increase the vascular flow. And also the secondary xylem is very strong because it has lignified cells. So it's going to provide a lot of strength as acting as wood. Okay. So that is done by the vascular cambium. Then there's another cambium, that is coke cambium, arising in the outer layers of cortex, sort of here. It is going to produce coke cell usually to the inside and the outside, right? So those coke cells have suberin in their wall, so they are going to die. Then they are going to produce the new dermal tissue replacing the epidermis that is being split, because why? New cells are being added inside, right? So because of that, you need a replacement. That replacement is called the periderm. What do you call the periderm? The coke cambium and the tissues it produces. That is all together is called the periderm. Okay, right? Then if you're talking about the vascular cambium initials, right? So in this vascular cambium orange color, you have initials that are going into the board. Those are going to produce the xylem and phloem sort of vascular tissues. And the initials that are sort of perpendicular like this, they are going to produce something called vascular rays that are actually connecting the xylem and phloem. Vascular rays. Any questions? Right. Come to the tute, Methana. We are going to do, we are going to write some things that are corresponding to each diagram. හරිද? First diagram එකට few sentences, second diagram එකට few sentences, and third diagram එකට few sentences. Because that's going to nicely follow secondary growth from the primary structure to the final secondary structure. Got it? තේරුණාද? Okay. I mean, this is actually the pith, right? It is the pith, but it's slightly reduced. Okay. Okay. Yeah. In front of the first diagram, right? In front of the first diagram, right? In a woody stem, write down in a woody stem, in a woody stem, vascular cambium vascular cambium is a continuous cylinder. Vascular cambium is a continuous cylinder of undifferentiated vascular cambium is a continuous cylinder of undifferentiated single cell layer located outside pith and primary xylem, located outside pith and primary xylem, and to the inside of cortex and primary phloem. Cortex and primary phloem. Okay, that is in front of this first diagram. Okay, top diagram. Then in front of the second diagram, right? In front of the second diagram, better draw this one. Okay, manage your space. Initials from vascular cambium are of two types. Okay, manage your space. Okay, වැඩිය පල්ලෙහාට එන්න එපා. Online people, in front of this middle diagram in page number 5, draw this chart. Initials originated from vascular cambium. If you want some understanding, if you can draw a single vascular bundle, and this is the parallel direction, this is the perpendicular to the axis. Okay, I will send a photo. What is the fate of the initials parallel? They become cells of vascular tissue, xylem and phloem. Okay. What is the fate of initials oriented perpendicular? They become vascular rays. Okay. Then in front of the third diagram, this third diagram, okay, cocambium arises in the outer layer of cortex, in the outer layer of cortex in the stem. Rises in the outer layer of cortex in the stem. Rises in the outer layer of cortex in the stem. Next point. Star mark at the right? Cocambium and the tissues it produces, cocambium and the tissues it produces are collectively called periderm. Are collectively called periderm. Okay, right? Called periderm. Mm, what else do you need to know? තව ඉඩ තියෙනවද තෙල්ලිය? No, right? Then fine. Right. Then there's something we didn't talk about. If you want to know the structure of a lenticel, I'll erase this one. Okay. Got it. Everyone got it, right? A lenticel is actually very, it's a simple thing, right? So those are like, if you take like a castle wall, right? There are small ventilation openings, right? There are, I mean, it's nicely protected by the periderm, but there are small openings here and there to get gas, to mainly to do gas exchange and also for to take some water in, all those kind of things, right? So a lenticel is kind of, your cork cells are nicely arranged in a box like, right? So if you take, this is the periderm, right? You have cork cells arranged nicely. And here, if you have in a place where you have a lenticel, the cork cells are sort of loosely arranged. And on top, you have sort of the remaining, you can say epidermis එකත් the remainder of epidermis. Right. So this is a lenticel. Okay. What is this? Period. This is like loosely arranged so that air and some water can come inside. What is the problem with these lenticels? What is the problem to the plant because of these lenticels? Organisms, pathogens, insects, fungi, bacteria, viruses can enter through these lenticels. They are an opening for infection, damage, right? So it's nicely protected, but chuti opening එකක් ඇති to go inside and harm the plant, right? So through lenticels, uh, pathogens can enter. So this is a lenticel. If you like, you can draw. Then in your page number 120, we have something small, bit of a definition that we didn't talk about. That is bark. What is bark? Singling එලය කියලා කියනවා. Bark එක. Right? All tissues outside vascular cambium is bark. All tissues outside vascular cambium. So, where is vascular cambium? This orange color layer. What are the tissues outside? You have your secondary phloem and a bit of primary phloem and periderm. That's it. Right? Bark is what? Highlight that. Bark in the resource book, highlight. Bark is everything outside the vascular cambium. That is the nice proper definition. Bark right? So that's the next thing we have to talk about. We'll draw a small separate diagram. I have given a diagram, right? Ah, yeah, in the next page you have another one. Page number 6 on top. Balanna, what are the bark? That sort of වරහනක් දාලා තියෙන what is shown as bark? Can you can see the vascular cambium, and what is outside the vascular cambium? Secondary phloem and periderm. That is bark. Got it? Highlight bark. Urandeva, right? Highlight. So you can draw a small diagram if you really want. What is this? This is the vascular cambium. Don't draw anything yet. Huh? We didn't draw it. Okay. What is this? Vascular cambium. Right? I'll explain this wood story to you right now as well. Right? So, as a plant grows over many seasons, there are growing seasons. This secondary growth happens in the nice growing season. Right? So, in the growing season, there is growth, usually secondary growth happening, usually, I mean, a bit higher, greater because it's a growing season, right? But in sort of, let's say, if it's a temperate country, there are clear seasons. There is a winter and a summer and a spring, like that. There are rainy season and a dry season, like that. In Sri Lanka also you have, right? So in the rainy season is the season for growth. The dry season is the season for just to survive, right? Just to survive, get through the season. So growth happens in the rainy season. So like that, there are growing cycles. Almost every year, there is a growing cycle. There is new layers of tissue produced every year. Okay. So as a plant lives for many years, sorry, a tree lives for many years, you can clearly see layers of wood, like layers of these vascular tissues and also wood that are layers and layers and layers. Have you seen when you are transporting wooden logs on a lorry, you can see like rings and rings of layers, right? Those are indicative of how old that tree is, right? How many years you can count and see, right? Because almost every growing season, you can roughly say every year, it's going to produce new layers, new layers, new layers, right? So the next thing we want to talk about is the different types of wood, right? What is wood? Secondary xylem. ඒක දැන් දන්නවා. Wood is secondary xylem. Okay. So this is vascular cambium. What is produced to the inside? Secondary xylem. So the most oldest secondary xylem is going to be called like a special wood that is called heartwood. Heart. It's like the center of the plant. Heartwood. Okay. And the most recent secondary xylem is going to be called, it's also wood, it is sapwood. Why is it called sapwood? Because that wood is still transporting water and minerals. So it's like wet again. It's still wet. But this most oldest secondary xylem is dry, yeah? Because it's not no longer transporting, it's dead and completely dry. That is called hardwood. And the still transporting secondary xylem is called sapwood. Those are both wood, okay, to the inside of vascular cambium. So we just learned what do you call everything outside to the vascular cambium? Yes, what is everything outside to the vascular cambium? Bark. Bark again. Everything outside to the vascular cambium is bark. So this is bark, right? This is orange color vascular cambium. Okay. Then there are two types of wood, right? The oldest wood is called heart wood. It's like the heart of the tree, like closest to the center. Then the still working xylem is called sapwood, bark and wood. Parts දෙකයි තියෙන්නේ for a plant. For in a tree, you have a bark and a wood, right? So when you are making furniture, what do you do those in those sort of mills? You remove the bark and you take the wood. From that wood also, ලාභ බඩු හදන්නේ මොකෙන්ද? Cheap furniture ඔය ලාභ ඇඳන් එහෙම තියෙන්නේ. Sapwood එකෙන්. They make the cheap furniture from sapwood. That sapwood is not properly hard enough. It's still working. It's still sort of, I mean, you can say it's the ලී ඇද ගහනවා කියලා කියන්නේ. Have you heard? Wooden furniture becoming sort of bent. That's because it still has water. Water is there in sapwood. Yes. So sapwood is cheap. You can make, if you make furniture from sapwood, it can be cheap, right? So what do you make? What do you use to make the most expensive furniture, most durable and the heaviest? Like you use hardwood, right? Hardwood එක තමයි අරටුව කියලා in Sing, right? So using that අරටුව you can make the most durable furniture. Therefore, it's expensive. Okay, right? That idea is there, no? Okay. So we have talked about the different steps of primary structure to secondary structure transition. Neither. So next step is to we have to draw this. So we drew the dicot stem primary structure, right? If you have space just underneath it, good, but can go to the next page. Huh? Online people, draw this. Always, while you are drawing, think what becomes with what becomes what produces what. Yes. And how do you start to draw this? Always start with the vascular cambium. Okay, listen now. When you are drawing, start with the vascular cambium, then draw what is produced by that. Okay, then draw the cork cambium and what is produced by that. When you are drawing, also go with the natural way, right? I think cinnamon is the periderm. It's not the bark, no. It's the periderm that we are removing. Kurundu potu කියලා ගන්න, it's the whole periderm. Because why? Okay, that's a good question. Kurundu potu. So this cork cambium produces cork cells. Usually, what is deposited in the cork cells? Suberin. But other sort of aromatic compounds. Have you heard of this sort of in chemistry when you're doing organic chemistry? Different organic compounds are there, lipid based, right? Aromatic compounds, remember carbon rings the compound, right? Other than suberin, certain other chemicals can be deposited in the cork cells. So cinnamon has a different aromatic compound deposited. That's why it has a taste and a smell and those things, right? But it is still potak, right? Got it? Don't draw the hardwood and sapwood thing, them. Only draw this one. Any questions? Okay, right? Online people still drawing? No? Okay. Then come to the resource book quickly. Come to the resource book quickly. Okay. Page number 121. Heartwood and sapwood. Heartwood and sapwood. Read. As a woody plant ages, older layers of secondary xylem no longer transport water and minerals. Right? As a woody plant ages, the older layers of secondary xylem no longer transport. Right? Why is it around? To be exact, there is actually a mechanism that is going to block these older secondary xylem from transporting. Right? There is a mechanism like across the what do you have at the end of xylem vessel elements? What do you have? What do you have? Uh, no. What do you have at the end of xylem vessel elements? Plate, perforation plate. Ne? Perforation plates. Yeah. Right. Those plates will become closed and transportation will be blocked across these older layers of secondary xylem. Right? There's a separate mechanism for that. So, old layers of secondary xylem no longer transport. Those layers are called heartwood. They are close to the center of the stem or root. Highlight heartwood. Okay. What is hardwood? Heartwood, older layers of secondary xylem, they are no longer transport. Then the newest outer layer of secondary xylem, still transporting xylem sap, is known as sapwood. Highlight sapwood. It's still sort of transporting the liquid, water and minerals, right? Sap. Then hardwood is generally darker than sapwood because of resins and other compounds that permeate the cell cavities and help the protect the core of the tree from fungi and wood boring insects, right? So the hardwood should be remain very strong because it's like the main support for a very tall tree. It's like a concrete එකම දාපු කම්බි වගේ, right? They should remain very strong. What is the commonest way that tall trees fall? කොස් ගස් එහෙම දිරලා වැටෙනවා කියලා අපි කියන්නේ, right? Tall trees, they decay while they are still alive. How is that? Where wood boring insects, termites, they drill through the layers and reach the core and they eat that. Therefore, the strength is lost and the tree falls down, right? In order to prevent that, there are sort of irritant chemicals in the hardwood that makes it more stronger, difficult to eat, and sort of maybe are repulsive to eat or something, right? So all those steps are taken to protect the support, right? Because every other tissue can be replaced, no, right? Every other tissue can be replaced, right? If you are damaged the phloem, you can replace. If the bark is damaged, you can replace. But if the core is damaged, the tree is going to fall down, death. No, there is no photosynthesis, right? Everything is going to be like cut in half again, right? Right? Got it? Only the young secondary phloem functions in phloem translocation and the old secondary phloem is sloughed off. Right? As the xylem increases, the secondary phloem also gets closer to the outside. So there comes a day the phloem also falls off and there's a new phloem in the inside. Okay. So in your come to the tute, right? Here. There's some space here, no? Tutti space here. Page number 5. Put a star mark and write down. Put a star mark and write down. Write it in a nice point form. Huh? Bark equals, write it as an equation. Bark equals what? Okay. Bark equals secondary phloem plus periderm. Bark equals, it's an equation. No? Bark equals secondary phloem plus periderm. Hurry. LA. That is bark. Okay. What is the other definition that you can give? Everything outside the vascular gap. If you want, you can write down that also. Bark equals secondary phloem plus periderm. Then what's something else you can write? Mammalian wood wood two types, heart and sap. Also you can say wood is equal to what? Secondary xylem. Right? Wood is equal to secondary xylem. Okay. Then heart, what are the things you can write down? Right? No longer trans, and you can say older, right? Close to the center. Sapwood, young. You can say still transports. And very, very important, the hardwood is darker. This is light. Sap food is light. Hurry. Hard food versus sap food. No longer transports. They are older. They are darker in color. They are located closer to the stem. Sap food. They are young. They still transport, and they are lighter in color. Yeah, root secondary growth will I'll explain next week. Huh? There's a question: is primary phloem included in the bark? Yes, I mean with age, primary phloem becomes irrelevant, right? It's very diminished. Adala naha, right? It's very small, and it's also sort of you can't even see it because you have dominating big layers of secondary phloem, right? So yes, technically primary phloem is included in bark, but we mainly say what secondary phloem and peridium. Got it? Next week I'll do root secondary growth. Okay. නැත්නම් ඒකයි මේකයි දෙකම කරන්න ගිහිල්ලා hard food sap food පැටලිලා bark පැටලිලා right because those things are not there in the root no root එකට එහෙම එකක් නෑ. Okay. Okay. Then okay. Then that's enough for today. Right? Homework. This page of the question tutor. Question number 7. There's only one question you can't do. There's this guttation killer part. Don't try that. But everything else. Page number 7 of the question tube homework. Okay. Page number 7 question tube. Structured essay. Do everything and come. Harry there. That's all for today. And also, if you want, you can draw this. But I have given you the diagrams in the tube. No need. Right. If you want your own diagram, you can draw. Do the homework after studying. Right. Try to do the homework Saturday night. Right. That's the best way. Online people, everyone has got the question tube. Question tube page number 7 structured essay is your homework. Any questions you have? So we covered a lot today. Definitely have to study. Primary structures of stem root done, secondary growth of stem done, and hardwood sapwood done. Big bulk. Right. That's it for today. Okay, send me a message if you didn't receive the tubes. I'll ask from the website. Okay. Okay. Right. Then thank you for joining. Okay. See you next week. Right? You can go. Okay, you're welcome.